Dear students, in this topic we shall discuss the inner ear cochlea. Cochlea is the coiled sensory structure in the inner ear that is specialized for hearing. It takes vibrations from the middle ear bones through its oval window and transforms them into nerve impulse. We shall discuss the structure of cochlea in detail. The cochlea has three fluid-filled canals or ducts along its length. These include, first, the upper scala vestibuli or vestibular duct. This duct is filled with perilymph. The second is the scala media, which lies in the middle. It is also called cochlear duct. It is filled with endolymph. And the third, lower duct, is called the scala tympani or tympanic duct. It is filled with perilymph. In this way, we can see that vestibular and tympanic canals are filled with perilymph, and in the middle lies the cochlear duct, which is filled with endolymph. The partition between the vestibular duct and the cochlear duct is called the Reisner's membrane, while the partition between cochlear and tympanic ducts is the basilar membrane. The cochlear duct contains the sensory organ which is specialized for hearing and is called the organ of Corti. This organ of Corti has hair cells which transduce the auditory stimuli into sensory signals. This organ enables human ear to detect and distinguish different frequencies of sound. We shall discuss the detailed structure of organ of Corti. It contains hair cells. It also contains on its lower surface a basilar membrane. It is supplied with auditory nerve endings, and on its upper side it has a tectorial membrane. There are two types of hair cells in the organ of Corti. These are the outer and inner hair cells. Both types differ in their functional anatomy. The inner hair cells are about 3,500 in number, and they are arranged in a straight line or a wide U shape. These hair cells are the true sensory cells which send impulses through auditory nerve. The outer hair cells are more numerous in number. They range in number from 12,000 to 25,000 in mammals. They are arranged in 3 or 4 rows and form a characteristic W shape. The outer hair cells have both sensory and motor elements, and they contribute to hearing sensitivity and frequency selectivity. Now we shall discuss the role of basilar membrane. Basilar membrane forms the floor of cochlear duct. ये cochlear duct के नीचे वाली तह बनाती है और जो पूरा organ of Corti है वो इसके ऊपर रखा हुआ है. This basilar membrane is the structure which is involved in the detection of sound according to frequency range. Now we shall discuss the role of tectorial membrane. Tectorial membrane जो है ये एक fibrous sheet है which lies on the apical surface of organ of Corti. जहां बेसिलर मेम्ब्रेन नीचे वाली तह बनाती है तो अपर वाली तह जो है या ऊपर वाली लेयर जो है दैट इज द टेक्टोरियल मेम्ब्रेन. टेक्टोरियल मेम्ब्रेन पर एक फाइन लेयर मौजूद है जिलेटिनस म्यूकस की. ये जो म्यूकस है इसमें जो नीचे स्टीरियोसीलिया हैं हेयर सेल्स के स्टीरियोसीलिया इस टेक्टोरियल मेम्ब्रेन के म्यूकस में एम्बेडेड होते हैं. नाउ द टेक्टोरियल मेम्ब्रेन जो है ये इंपॉर्टेंट रोल प्ले करती है उस वक्त जब बेसिलर मेम्ब्रेन इज डिस्प्लेस्ड. जब बेसिलर मेम्ब्रेन थोड़ी सी मूव करती है बिकॉज ऑफ साउंड वाइब्रेशंस तो ऐसे में टेक्टोरियल मेम्ब्रेन मूव्स ऑन द टॉप ऑफ हेयर सेल्स. ये हेयर सेल्स की टॉप के ऊपर मूव करती है इस तरह से. एज ए रिजल्ट इट प्रोड्यूसेस ए shearing फोर्स. ये shearing फोर्स प्रोवाइड करती है और स्टीरियोसीलिया ऑफ हेयर सेल्स जो इसके म्यूकस लेयर में मौजूद हैं उनको मूव करा देती है, वो अपनी जगह से हिलते हैं. द बेंडिंग ऑफ हेयर सेल्स generates graded potential. ये जो graded potential है that is transmitted to the sensory axons of auditory nerve.